And let's start that practice test. By clicking, you confirm your computer meets the testing requirements below. Yeah, we have read this. Sure. All right. It says the length is 47 minutes. I guess it's an online exam. All right, no next button. Oh, it's down here at the bottom. Okay. Unstable traffic flows occur when drivers accelerate and brake too often. Unstable traffic flows waste energy. The table below gives one example of traffic speeds in kilometers per hour in an unstable flow situation. Okay, so these are in every 10-second intervals. Let's see what the question is. What is the mean speed in kilometers per hour in this situation? All right, now because I know what I want to say mean equals average, but because median is middle, so we're just going to Google what does mean mean. Okay, what is the mean of this number? Will be 9, which will be the middle. The sum of all the observed. We know to find the mean of the given data, we need to divide the sum of all of the observations by the number of observations. So mean is average. So we'll just say, and then we're going to go, because I'm lazy, we're going to go equal average. Yep. Okay. So mean is not the median. The mean is the average. All right. So you may use the calculator. Oh, cool. It's the calculator I own for all my scientific needs. So we're going to go 30, 20, 50, 70, 30. Well, can I use the keyboard? 20, 50, 70, 20, 70, 20 equals. And then we have, what is that? 7 entries. I'm going to divide by 7. And it's going to give us 40. Boom. And then is there a way to check it? Just hit next. I guess there's no way of checking it. The cell cycle can be divided into 2 phases, interphase and mitosis, cellular division. Mitosis is further subdivided into prophase, metaphase, anaphase, and telophase. A lab technician observed 200 cells from a cell line and recorded the number of cells in each phase of the cell cycle. The results are shown in the table. So, based on the data, what is the probability a randomly chosen cell will be observed undergoing cell division? So, we don't need any of this information here. Basically, we're going to add them all up and then divide the whole by whatever phase they want. So you may use a calculator. Well, no shit, Sherlock. Mitosis is all of these. So basically, yeah, prophase, metaphase, anaphase, telophase. Oh, can't highlight them all, but these are our mitosis. So we'll just open up the calculator again. Well, thank you very much. Clearing the memory. We're going to go 170. Really? Fucker. Run 70 plus 20 plus 4 plus 2 plus 4. Gonna be... Oh. Can we use delete to clear it? Clear. So 170 plus 20 plus 4 plus 2 plus 4. Okay, it's 200. Now, what percentage of the cells are undergoing mitosis? We know that we're looking at 30. 20 plus 4 is 24, plus 2 is 26. 26 plus 4 is 30. So we'll just go 30 divided by 200 times 100. To give us 15% of the time the cells are. Another way you can double check this is 200 multiplied by 0.15, which is the decimal representation of 15%, will equal 30, and that's how much numbers we have here. All right, we have a word problem next. Scientists today are studying tidal power as an alternative energy source for generating electricity. Some scientists conclude that tidal power is a good alternative source of energy because it uses natural processes. Does not rely on fossil fuels, means it relies on the turning of the earth, and does not release greenhouse gases. These scientists recommend building tidal power plants around the United States in locations with high tidal power potential. So that means in the oceans around the country. Other scientists conclude that tidal power is not a good alternative energy source because tidal power plants can negatively impact the surrounding ecosystem by killing marine animals, restricting fish migration, and inducing the natural flow of water and causing silt buildup in waterways. These scientists recommend not building tidal power plants in U.S. waterways. Oh my God. Both conclusions are based on valid data and scientific reasoning. How can both conclusions be valid? All right, well, first question is, two groups of scientists believe that different theories about the process of generating electricity. Theories doesn't mean you hypothesize. Hypothesize means hypothesize. They have theories, which means they have facts to back up their thoughts. That could be true. Two groups of scientists are looking at different specific aspects of the same scientific problem. The two groups of scientists collecting and analyzing data about all types of possible alternative energy sources. The two groups of scientists are on opposite sides of the debate on whether the United States needs to find clean. Well, this one's automatically wrong because they are both looking at green energy, it looks like. Other scientists conclude that tidal power is not a good alternative energy source. We're talking about alternatives, so this is automatically wrong because they're not talking about fossil fuels, they're talking about clean energy. The two groups of scientists collect and analyze data about all types of possible energy sources. Well, we're not talking about that. We're talking about clean energy, so that's probably wrong. The two groups of scientists are looking at different specific aspects of the same scientific problem. That's true. The two groups of scientists believe different theories about the process of generating electricity. It's not about the process of generating electricity. It's about the downsides. So B has to be it. I hope I got that right. Read this paragraph about the benefits of a green roof. Oh my gosh. Which sentence from the paragraph supports the idea that green roofs can help reduce cooling costs? So now we just need to find the sentence about cooling costs. Scientific research has shown green roofs has many benefits compared with traditional roofs. I think that was pre-stated. Plants naturally absorb energy from the sun and convert it into chemical energy that is stored in the bonds of sugars during photosynthesis. That's true. The soil material also helps insulate the building. In addition, a green roof helps control runoff as plant roots absorb rainwater. Which sentence from the paragraph supports the idea that green roofs can help reduce cooling costs? Soil material helps insulate the building. It's the only one that talks about temperature. Insulation is about regulating temperature. This is about water runoff. Plants absorb energy from the sun and convert it to chemical energy. This is just talking about photosynthesis. Has nothing to do with insulation. Scientific research has shown that a green roof has many benefits compared to traditional roofs. Does not talk about cooling costs. Oh no. All right, let's go to the end here. During a drought, the decrease in rainfall will reduce the amount of vegetation that can grow and increase the occurrence of natural fires. So an elevation in blank and a reduction in blank should be found in the cores. Pollen sediment wetlands. Charcoal pollen sediment wetlands. Okay, so this is probably talking about historic information. During a drought, the decrease in rainfall will reduce the amount of vegetation that can grow. Right? So we have a drought, we have less vegetation in the core sample, which is a borehole. Let me see here. Let's pop over here real quick. Core sample, and so you have the ground. Oh, you have the ground. They have a drilling rig. We'll put a little fucking corkscrew there. They drill down, and then they pull out this section here. And then depending on what's in each layer and how far down it is, they can tell how much time. The further down, the further back in time you go. So they know that so many feet down, let's say 100 feet down, they know that right here was 1,000 years ago. I'm just using a random number. So they know 1,000 years ago, if they see less plants, right, they know that it was generally a drier period of time. And then we'll come back over here, back to our tests. So we know what a core sample is because I figure you might have some need for that. It's probably in here. No, it doesn't look like they do. So during a drought, decrease in rainfall reduces the amount of vegetation that can grow and increases the occurrence of natural fires. So you also find burnt plant matter in those layers on the core sample. So an elevation in charcoal and a reduction in pollen should be found in the cores. Let's come back through here. Sahara Desert and the Nile River are two components of Egypt's natural environment. In Egypt, 90% of the population lives in only 5.5% of the total area along the Nile River, residing along the banks of the Nile. See, this small percent represents the only farmland in Egypt, but it's called the fertile Nile for a reason. Annual flooding of the Nile supplies water for crop production. Without this water source, civilization in Egypt would be very different. Records indicate that a drought caused the demise of Egypt's old kingdom. Because of an insufficient water supply, food production decreased and population suffered from famine, and the political system destabilized. Scientists have recently investigated whether other evidence supports the theory that a shift in climate led to political upheaval in Egypt about 4,200 years ago. One group of researchers removed a sediment core from the Nile Delta. The layers within the sediment core allowed researchers to investigate environmental conditions over thousands of years. The researchers hypothesized and observed that periods of low concentrations of pollen from wetland plants and elevated concentrations in microscopic charcoal would occur together, indicating a decrease in vegetation and an increase in natural fire during drought years. So, occurrence of natural fires saw an elevation in charcoal and a reduction in pollen. Here's your pollen, and there's your microscopic charcoal. Should be found in the cores. Yeah. All right. What else should the second group of researchers measure? What else should the second group of researchers measure to test its hypothesis? The current level of water in the river, the type of pollen in cores from a non-drought period, the composition of microscopic charcoal released during natural fires, the quantities of sediment in the cores from the delta during a non-drought period. Let's see what this last thing says. A second group of researchers also examined sediment cores by analyzing shifts in the amount of sediment decomposed over time. These researchers hypothesized that a decrease in rainfall would correspond to diminished vegetation cover, which in turn would lead to an increase in erosion. So sediment is sand and that's transported by rain. So think of like the shore of a lake or a pond. You have really fine sand like mud. That's sediment. This increase in erosion represents drought-like conditions and could be observed as large deposits of sediment within the cores. What else should the second group of researchers measure to test its hypothesis? The quantities of sediment in the cores from the delta during a non-drought period. The composition, nope. Of microscopic charcoal, we don't want that. That's for fires. Types of pollen in cores from what does that have to do with sediment? Current levels of water in the river. Might, but this is really what it is. Okay, I'm not seeing any chemistry yet, so I think we're off to a good start. And we're almost halfway through. Scientists often experiment with brassica plants because brassica plants grow very fast and they produce seeds 30 days after being planted. The quick maturation, which is maturing, allows scientists to study several generations of brassica plants in a short time. Usually the scientists use brassica plants to study genetics and inheritance. Pea plants have generally been used too. That's where we get the Mendel plots, which is what this is. So probably brassica is a pea plant. In one investigation that studied the inheritance of brassica stem color, scientists crossed a heterozygous dominant. So RR with, okay, big R is a dominant, little r is a non-dominant gene. Plant with a homozygous recessive. So recessive means that both are little r's. And then, homozygous means both, homo together. So homozygous dominant would be big R's, recessive would be little r's. The dominant and the recessive are implied by the upper and lower. So heterozygous genes will be the big R's. This results in offspring with purple stems, RR or big R, because anytime you have, these are genes in your genome. You have sets of chromosomes and you have genes that make up the chromosomes. So anytime you have a dominant, it's a big R, and if it is heterozygous, anytime there's a big R, you will get the gene from the big R. If it's a recessive gene, so certain physical traits in your body, like your eye color, your hair color, not necessarily those eye colors, you get a variation. In hair color, you have a variation. So big R, little r lists, there's two sets, and then so you take your parent set, or one parent set and the other parent set, and you can get a variation. That's how you can have people that have like blonde hair, brown hair, dark brown hair, and black hair. And those kids will all be produced by the same parents because it's a blend. And then some genes, it's just one from dad, one from mom. And if it's a heterozygous, the larger case gene will override all of the other gene data. So this resulted in offspring with purple stems that are big R, or big R, little r, because it's a dominant gene. So it doesn't matter if it's two bigs or one big, one little. You're not going to get the opposite phenotype, which is the expression, like blue eyes or green eyes is the phenotype of the gene, unless there's two recessives. two smaller ones. And offspring with green stems, you see, what I just talked to you about right now, this resulted in offspring with purple stems if there's any uppercase R's, and offspring with green stems if both parents had recessive R's. So that means there's 4, 2 from one parent, 2 from the other parent. All 4 have to be little r's for the recessive gene to be dominant. A Punnett square, that's a Punnett square, sorry, for this cross is shown below. So the way this would work, I'm going to have to come over to the other side here. We're going to come over here, and I am running out of time, so we might have a hard time here. We're going to erase this real quick, and we're going to do a Punnett square. And so come over here. A Punnett square is just a square, right? We make a square, and if we have a big R and a little r, 2 little r's, it doesn't matter what group is here and what group is here, as long as these pertain to one parent and these pertain to the other parent. And with plants, you might go, there's no parents. Well, there actually is, because there's one plant that is producing pollen and one plant that is going to produce seeds, which are the babies. So think of a nasty analogy is the pollen is cum and the seeds are the babies coming from the mom. So the way we do a Punnett square is dad produces this big R, mom produces the little r, dad produces the big R, mom produces the little r, and then dad produces little r, mom produces little r, little r, little r. So what this means is out of 4 children produced, 2 will be gene type big R, little r, and 2 will be gene type little r, little r. So out of every 4 offspring, you'll see a 50-50 mix of the children being dominant gene and recessive gene. Okay, and now, so this is where it gets weird. If we were to do this again, right, and switch these two let's see here. We want both parents to be the same, right? So big R and little r. What we end up getting is big R, little r, and then we end up getting R, little r, little r. So that means out of the 4 children that produced, you have 75% will show the dominant gene and 25% will show the recessive gene selection. So think of this as brown eye color, and then this one as blue eye color. And this is very similar to how brown and blue eyes work. That's why you have brown eyes, your mom has brown eyes, I have blue eyes, Grant has brown eyes, or hazel, but they're brown. And that's because you need the recessive gene to have that expression. So what that means is that to have blue eyes like I do, I'm 1 in 4 unless both parents show the recessive, homozygous recessive. So that means both are the same. So that means out of a Punnett square, if they're both little r's, the expression of someone's genes will be the recessive type. So 100% of the time with blue eyes, right? It's not exact with blue eyes. I have to look it back up, but I'm just using this as an example. 100% of the time, the children will show up with blue eyes. So now that we kind of have a primer on how a Punnett square works, which this is basic, it's not chemistry, it is biology. You may use a calculator. What type of answer in the boxes? What percent of the offspring will have green stems? Well, if we just did a Punnett square, right, where it's big and little, little, you know, this box is going to be big, little. This box is going to be little, little, right? This box is going to be big, little, and this one's going to be little, little. It's the same thing we just did. They were just flipped, so these were on the left side. So that means 50-50. So what percent of the offspring will have green stems, right? And what percent of the offspring will have purple stems? Right? So we know that R can only come in these, big R can only come in these boxes, right? So then these boxes will always be little r's. So we're going to say 50. We're going to come over here and we're going to say 50. Now I can see if you've never seen this, it would be very hard to kind of figure out off these what's going on. Okay, peat is a mixture of partially decomposed plants and animal material found in waterlogged, acidic, anaerobic environments. Anaerobic is a very good term when dealing with water. It means oxygen-rich water. Which conclusion can be drawn based on the information of the table? So let's not even read this, right? Because we don't really, we want to figure out what conclusion can be drawn from this table. Okay, so stages of peatland restoration. So re-wetting makes surface wet again. It takes about 3 to 5 years to re-wet the ground. Renaturation, which is basically nature and ation. It seems like remaking it filled with nature again. Replaces vegetation. Oh, it gives you a description there, right? So you kind of have a feeling that that's what it means, and then it tells you. It takes several decades for the natural environment to come back. Regeneration accumulates new peat. So it has to be partially decomposed plant matter in a waterlogged, acidic ground. So depending on what's in the rock and dirt, you can have it be basic or acidic. So it needs to be acidic, so it needs to build up the acids, probably from decomposing matter, and it needs to have high oxygen, so it takes 100 years or more. Which conclusion can be drawn on the information in the table? Peat is a fossil fuel. Nope. Peat is a fossil fuel. Peat is not a fossil fuel because it has biological origins, making it a biofuel. Peat is not a fossil fuel because humans can promote the replenishment of peat. That sounds like the right answer, right? So somewhere around here in this thing, it's going to be talking about fossil fuels. To be considered a fossil fuel, a material must be biologic in origin, be used as a source of energy, and replenished on a timescale greater than human lifetimes. Peat reserves have declined over time. However, peat harvesters claim that restoration processes can make peat a renewable resource and allow it to be classified as a biofuel. The restoration process is outlined below. So 100 years is kind of a little bit more than a lifetime, but fossil fuels, fossil is in the name, right? And fossil implies millions of years, if thousands at minimum, right? Tens of thousands. Which conclusion can best be drawn from the information of the table? Peat is a fossil fuel because rewetting it only... That's bullshit. That makes no sense. Peat is a fossil fuel well, what did I just say? Fossil, right? So we're going to disregard the first two. Peat is not a fossil fuel because it has biologic origins, making it a biofuel. Right here. Must have a biological origin. So that's wrong. So peat is not a fossil fuel because humans can promote the replenishment of peat. Yeah, sure. We're going to go with that. That doesn't make any sense. Maybe it does. Why do individuals with spinocerebellar ataxia experience loss of muscle control, even though the ataxin 1 protein builds up only in the brain and spinal cord? Inclusions can build up in all body systems and cause debilitating effects. That sounds right. Genetic mutations in the ataxin 1 protein cause muscular proteins to change shape. When the ubiquitin tagging system does not work effectively, cellular waste can build up in the muscles. builds up only in the brain and spinal cord. Why do you experience muscle loss control? That's starting to sound right. Tagging systems in order to effectively so their waste can build up in the muscles. Neurons affected by the toxin 1 protein buildup are in the region of the brain that manages muscle. Oh, B, D, and C really look good. Okay, is Alzheimer's disease, which are a small polypeptide chain, about 76 amino acids long, are called ubiquitin. Okay. Proteins are no longer needed. Okay. A cell must continuously sort useful material from its waste products and remove or recycle the waste. One way a cell does this is to first put tiny tags on proteins that are no longer needed. These tags, which are small polypeptide chains, blah, blah, blah. Ubiquitin tags direct proteins to compartments of the cell called proteasomes. Within each proteasome, ubiquitous tagging proteins are deconstructed into component amino acids. These pieces can be repurposed to build new proteins. That is in the... Oh my god. The large blank. Sometimes organisms can process waste effectively, allowing waste to pile up and contribute to many diseases. Alzheimer's disease, kind of, it's more of a lack of transfer of certain chemicals. Includes piles of waste proteins in the brain called tangles, which Parkinson's disease involves the accumulation of recyclable material into Lewy bodies. Oh my god. Yeah. I'm having PTSD from biology. Okay, tagging 1, accumulate, which leads to cell death. The cell's most effective vegetation are neurons and part of the brain associated with motor control and spinal cords. So a person with immunity 1, I'm going to call it AT1, progressive loss muscle control of these cells. So, which one was it builds up in the brain that manages muscle control? Boom. There's like no science. What statement describes the overall consequence of proteins containing mutations like these described in the passage? Overall consequence. Okay, waste will build up and will resemble a new build. Resemble to build. No, waste will build up, and Mallory bodies will accumulate. No, wrong. Will eventually be exercised. No, wrong. Will build up in vital organs, and the organism will experience disease. That sounds about right, right? Based on the sample set of data, which statement correctly identifies a weakness of the sampling technique? Sample sites were not selected based on the range of crater densities. Some missions took samples that were known to be very different ages. Samples were taken from both dark and bright lunar areas instead of concentrating on one area. The missions did not sample craters on Earth, where rocks were more easily accessible for radiometric dating. So the graph below compares sample ages to crater densities from each landing site. So ages based upon density. Matchmaking radiometric dates to crater densities creates a scale for estimating the age of any visible region on the moon. Oh, fuck you. Accumulate over time, so older regions of the moon's surface are more craters than newer regions. Well, of course. Sample sites were based on range of crater densities. Well, that's the whole point. Some missions took samples that were known to be very different ages. Samples were taken from both dark and bright lunar areas instead of concentrating in one area. The missions did not sample craters on Earth, where rocks are more easily accessible for radiometric dating. That has nothing to do with the moon. The dark and light, I'm not getting why it would matter. They're purposely taking information from different places. They say that sample sites were not selected based on a range of craters. Oh, they were not. After a meal, glucose is absorbed from the small intestine, starting the following process. Blood glucose increases, pancreas responds, secreting insulin into the bloodstream. Insulin causes the liver to convert glucose to glycogen, reducing blood glucose. The pancreas stops secreting insulin. Low levels of glucose cause glucogen to be released. Glucagon. Glucagon causes the liver to break down some of its stored glycogen to glucose, increasing blood sugar glucose. The graph shows these changes over a 9-hour period. At point A on the graph, blank is turning to blank, right? So point A on the graph, this period during the increase, glucose is observed. And then once we get down to here, it looks like this is where we're going to turn glucogen back into glucose. Glucogen into glucose. And then we're going to go, okay. And then based on the table, how many days will the researcher experiment take? A young researcher wants to test how well extremophiles, organisms that thrive in extreme conditions, live in different environments. She will do this by putting extremophiles of different species into different solvents. Because of limited lab space, she can only run one sample at a time, and each sample must remain undisturbed for 7 days. To reduce the total time needed to process all the samples, the researcher decides to use 2 acidic solutions along with a control. This information below includes the extremophiles and the solvents the researcher will use to test. Looks like there's 3. Here's the control. Here's the 2 different acids. So she can only run at a time. So 7 days for each one. There's 3 tests for each one of these bacteria. 1, 2, 3, and 4. 4 times 3 is 12. 12 times 7 is 86. Is it 86? 12 times 12, 7 is 70 plus 14 is 84. It is 2 in the morning. How many reagents? Fuck this. Which change to the experimental design would improve the reliability of the engineer's measurements? Using liquid other than water. Using liquid other than water to determine porosity. Using flasks instead of beakers. Testing single samples for more than 3 locations. Testing more samples from each location. We got 6 minutes to do 3 questions. We're good. A group of civil engineers wants to determine whether the porosity of surface soil affects aquifer recharge rates. So porosity is just little holes that space in between rocks. And water molecules, just the H2O, can go through those little pores. Pores are just holes. The ability of aquifers to refill with water. The engineers conducted the following study to determine the porosity of the surface soil at various locations. The results of the study will then be compared with the recharge rates of the aquifer each location supplies with water. Collect 1 200 ml sample of soil from each 3 locations. Place each soil sample in a beaker. Add 200 ml of water in each sample and allow the water to saturate the soil. Measure the amount of standing water in each beaker. Amount of water the soil did not absorb. Determine the amount of water in the soil sample by subtracting the amount of standing water from the 200 ml sample, dividing the amount of water in the soil by 200 ml, then multiplying by 100 to determine the percent of porosity which change to the experimental design would improve the reliability of engineers' measurements. In any type of scientific thing, to get better improved measurements, you increase the amount of measurements. So it doesn't matter if you use a beaker or a flask, they're still tubes. Testing samples from more than 3 locations could help. Testing more samples from each location. I'm going to say we're going to test more locations. Because it sounds like they got this down to a science. So testing the same soil twice, I don't think it's going to help. But testing 100 feet over and doing it again, I think will definitely help. Actually, hold on. Let me make sure I read that right. Testing single samples from more than 3 locations. Okay. Porosity of the surface soil at various locations. Results of the study will be compared with recharge rates of the aquifer at each location. Oh, so we don't want samples from not those 3 locations. So we're going to just do more samples. Maybe a foot over. Which comparison of the new process of hydrogen production and photosynthesis is supported by the information? Which comparison of this new process of hydrogen production and photosynthesis is supported by the information provided? The new process is more efficient than Okay, I actually have to read this one. Found a new way to make inexpensive hydrogen that can be used as a fuel. The process of hydrogen production has some similarities with photosynthesis. In plants, photosynthesis uses energy from the sun to chemically combine water and carbon dioxide into sugar and oxygen gas. The sugar is then used as fuel source for the plant and organisms that consume the plant. Right, because it's a primary producer, which was a question in one of your other tests for science, because plants are primary producers. To produce hydrogen, scientists mix extremely small particles of cadmium and selenium, which emit electrons when exposed to light, and absorb a gas and a chemical that produces hydrogen ions when decomposed. The scientists use a nickel catalyst, which causes emitted electrons to combine with the hydrogen ions and form hydrogen gas. This reaction is continuous, and after 2 weeks, the hydrogen gas was still being produced. The scientists hope that this will be a viable fuel source in the future. Which comparison of this new process of hydrogen production and photosynthesis is supported by the information? The new process is more efficient than photosynthesis. Both processes change light energy to chemical energy. Both processes result in the production of hydrogen gas. Wrong. Only this thing does. Plants don't make hydrogen gas. The new process emits more electrons than photosynthesis. Wrong. Right? There's nothing in here that says this. Okay. To produce hydrogen, scientists make extremely small particles of cadmium and selenium, which emit electrons when exposed to light. And absorb acid. A chemical that produce hydrogen. The scientists use nickel catalyst, which causes the emitted electrons to combine with hydrogen ions and form hydrogen gas. This reaction is continuous after 2 weeks. No, it's just the process changes light. That's how they're the same. With comparison, they just use light as the chemical catalyst. Last question, 1 minute, 38 seconds left. What is the independent variable in the experiment? So, a group of scientists studying the environmental impact of internal combustion engines burning hydrocarbon fuels. The scientists equipped 4 vehicles with devices to capture measure particle emissions. 1 vehicle burned diesel fuel, 1 burned ordinary gasoline, 1 burned gasoline ethanol mixture, and 1 burned natural gas. The 4 vehicles had equal masses and carried identical cargo. The scientists drove each vehicle 400 kilometers, recording the volume of fuel burned and the quantity of particle emissions generated. What type of independent variable? What is the independent variable in the experiment? Mass of the vehicle, quantity of. This is the independent variable. So we have 46 seconds left. Let's come over here to the drawing board. All right. So over here on the drawing board, we're just going to run this through. Independent variables. We're going to have a graph here, X and Y, right? X and Y. So the independent variable right here is how much is produced on the x-axis here. So we're going to say 1 mile, 2 miles, and 3 miles, right? Let's say we're producing 0 emissions. And then as we go 1 mile, the emission goes up. And then we go another mile, the emission goes up, right? And then on another graph, what we have here with these is with this other fuel source, as we go 1 mile, the emissions are lower. The emissions are lower. So this is the value that is changing. The fuel source for each vehicle doesn't change. So that's an independent variable. The miles are independent variables. The only thing that changes in the graph is going to be the y-axis, and that makes that the dependent, because y depends on the value of x. So whatever the f of x is going to be will give you your y. Actually, it's the other way around. It's f of x will equal x. And then this will be your y. And so whatever is on the x side of the equation will dictate whatever the y is. So whatever you're putting on the x will give you a y. Very confusing, I know, but just remember that the y-axis is going to be your, depending on how you set it up, it's going to be your dependent variable. So let's come over, pop this back on, hit OK, and let's see how many time has expired. Your time has expired. Click OK to continue. All right. There is no score report for this exam. What do you fucking mean, motherfucker? Oh no, I'm unemployed. Thank you for setting up your profile. Fuck you very much. Congrats. You're ready to take the GED test for science online or in person. Not yet. Does it tell me my score? Does it tell me what I got wrong? View score report. Oh, 170, likely to pass. Let's pull up the calculator real quick. And there was, what, 16 questions divided, well, 16 questions divided by 200. 0.08. $170 divided by 200, going to be 85%. So oh yeah, 200 divided by 16 divided by, it's 12.5% per question. So what is that? We missed one question because of it's 12.5% per question. And we got an 85%. No, we missed two questions. $170 divided by 200 is 85. $16 times 0.85. 13. So yeah, we missed two questions. Because it would have been 14 out of 16. So, fuck us. I guess we don't get to know what we got wrong. Stacking the deck. Alrighty. How do I stop this? Alright. I hope this helps.